Hi, my name is Will Krawczyk, Scientific Services Manager at Chemical Computing Group. Today I'd like to show you how we can model a mouse antibody and inspect it for liability motifs. We'll then model a panel of 14 candidates, also inspect those, and then choose the most promising in terms of improved developability. So let's get started. So first let's start by modeling our mouse antibody. So I'm going to go ahead and open up the sequence using the open button, and then we'll just retrieve the file that we're going to be working on. So this is a FASTA file. It contains only a sequence. You can see it's quite a small file size. And when I click OK, Mo recognizes that that's a sequence, and for that reason it opens it up directly in the sequence editor. The sequence editor is our second major window type in Mo. We use it to display sequences. It's also our launching point to the various protein modeling tools that we have. So I'm just going to adjust the view settings on the sequence editor by first wrapping the sequence, and then secondly I'm going to switch to the single amino acid code rather than the 3-letter code. And here we find that we've got our two chains. We've got light and heavy. And next I'd like to annotate the antibody. So I can do that directly from the sequence editor. Here we find some of the default themes available to us, such as IMGT and Kabat. Today though we're going to be using North, and when I click North you'll notice a couple of changes. So firstly, it split our sequence into different panels. By default our light chain is displayed in the leftmost panel, subunit 1. And then we get our heavy chain shown in subunit 2. You'll notice that the sequence editor has been, the sequence itself has been colored to show us the CDRs, the complementarity determining regions. So we've got L1, L2, and L3 for the light chain, H1, H2, and you'll notice that the third CDR of the heavy chain has been colored red. This shows us that it's more variable than any of the other CDRs. Now that we've got our sequence display set as we'd like it, we can move on to create our model, and we can do that again directly from the sequence editor. We've got a couple of options for generating models. We've got a homology modeler, but today we're going to be using the antibody modeler, which has been created specifically for antibody modeling. So the panel itself has been designed so that we can open it with the sequence loaded in Mo and just go ahead and click Build to start that build process. The application itself comprises 3 main tabs. The Model tab allows us to select the sequence that we'd like to build. It also gives us options for the type of model that we'd like to make and how it should be annotated. The second tab lists the potential frameworks to be used, and the third tab lists the potential CDR loop candidates. So in this case, we're modeling the chains in Mo using the sequence mode. We're going to be creating an FV model having the exact same sequence as the input. So I'll just go ahead, click Build, and I get a message at the top left of the main Mo window. It gives me a progress update on that modeling process. We've also got a database viewer which has been opened where the results will be written. In the background, Mo is now searching for the appropriate template to use, and the first task in antibody homology modeling is to find a suitable framework that we can use as a structural core. So Mo's searching the antibody database for structures from the PDB that contain a variable domain, and the promising results are written to the Framework tab, along with their PDB code and a score. The scores are based on a combination of sequence identity and structural quality. The hit list is sorted by the average score for both the VL and the VH frameworks. Beyond this automated workflow, I can control and manually select the templates, and I can select different templates for the different chains. In this case though, I can see that one SY6 has been selected on the basis of it being over 92% identical to the query framework. By the way, I see that the modeling process is complete and a model has been written to our output database. But before we look at that, let's just finish looking at how the modeling process advanced from the framework. So once we've got our framework, we then searched for the CDR loop templates. Again, Mo is searching the PDB for CDRs that have the same length, the highest sequence similarity, whilst avoiding clashes. The template that's been selected has got a plus next to it, and that process is repeated for each of the CDRs in first the light chain and then the heavy chain. The 6 selected CDRs are then grafted onto the framework, and again I can customize which templates are queried to leverage my own structures and have complete control over the selection of each CDR. The final result is written to the database viewer. So we now have our mouse antibody model, which we can call our wild type. But in the next part of today's session, we're going to be analysing a series of antibodies to identify the most promising for improved developability over this wild type. So before I get into analysing this first model, let's start the modelling process for the 14 candidates. So I'll go back to the Model tab, and instead of modelling from the chains that we have in Mo, I'm going to model from a file. I'll select the file from the sample mol directory, and this file contains 14 sequences. I'm just going to change the name of the output database, and then I'll click OK to start that run process. I get a message at the top left of the main Mo window. I'm just going to let that run in the background. So let's go back to our wild type, which is displayed in the third major window type in Mo. This is the database viewer, which looks and feels much like an Excel spreadsheet, but if I open this field, you see that it actually contains the 3D structure of our model. We can also use this as a launching point to be able to leverage protein engineering tools such as the ability to calculate protein properties. In this case, the database viewer displays our single model that we requested to be written to the database, together with a query sequence and the templates that have been used to generate this model. I'm just going to send that model directly to the main Mo window by double-clicking, and I'll click yes to clear the data, and here we see our finished model. You'll notice that the rendering scheme has been automatically extended to the model. So in this case, we used North, whereby the CDR regions were colored purple, so we can identify our L1, L2, and L3. You'll also notice that the light chain has a slightly lighter shade, whereas the heavy chain has a slightly darker shade. You'll also notice H1, H2, and the red H3. Also notice that the sequence editor and the main Mo window are synced. So if I make a selection of atoms in the main Mo window, they become selected in the sequence editor and vice versa. So next, what I'd like to do is open up our system manager, and I'll just pin that to the main Mo window. The first thing that I notice here is that we've got a single line for our receptor model. So what I'm going to do is just switch the view from basic to extended, and then that provides complete control over these light and heavy chains separately. The reason I've done that is because the system manager provides a convenient way to control the visualization of the different proteins in the system. So from here we can hide and show different atom sets, we can make selections, we can also render and generate surfaces. You'll notice that we also have this item called sets. If I click to expand, you can see that when our antibody was annotated, it's created these sets or collections of atoms, which allows easy selection of these sets so that we can use these when we run various applications across Mo. Now that we have our model, we can start to analyze the properties and think about potential issues with respect to biophysical properties. So for that, I can generate some protein patches. And the reason I've selected protein patches is because it shows me the important regions on the surface that have either excess charge or hydrophobicity. The hydrophobic patches are colored green, negatively charged patches are colored red, and the positively charged patches are coloured blue. Large hydrophobic patches, such as this one here, can be responsible for binding other proteins and can cause us issues for self-association or aggregation. Charged patches help binding via salt bridges, but they can cause us issues with respect to viscosity. In this case, we see that we've got a hydrophobic patch and a negatively charged patch, both surrounding the CDRs. I'll just clear our surface, and beyond the biophysical properties, we can also look for potential liabilities. And one such liability might be the discovery of a glycosylation site. We can identify these from a sequence pattern and the exposure of residues. So within the sequence editor, we actually have a selector, and we can use the selector to leverage Mo's selection language to quickly select residues or motifs using keywords and expressions. It can easily identify exposed sites like oxidation-prone methionines. In this case, though, we're going to be searching for a glycosylation site using a prosite motif. So if I click on the pull down, you'll see some of the predefined options available to us. So I'm going to use the prosite motif with a note on exposure. I'll click enter, and I see within the sequence editor some residues have been selected, which indicate that a possible liability has been found. Next, I'm going to render them so that they're easily identifiable. So I'll render both the atoms and the residue. Perhaps what I'll do is show these in the main Mo window, and then I can also add a residue label so we can keep track of which residues these are. So here we've identified a liability motif and selected those associated atoms in 3D, and we find that these are all solvent exposed. We know that asparagine and serine are essential for the glycosylation motif, and because serine has a slightly lower potential of interacting with the antigen due to its position, we're going to identify substitutions for the serine position to remove the glycosylation liability. But the question then becomes, what residue do we want to mutate to? One possible way to answer that question is to look at similar structures and see what residue is commonly found in that position. For that, we can leverage the Mo antibody project, which has a set of curated antibody structures, all aligned, superposed, and annotated in a consistent way that allow easy comparison for tasks exactly such as this. So I'll go ahead and open up the antibody search application, which is the interface to the Mo project database, which contains public domain antibody structures. There are two sections to this panel. At the top, we get a list of the antibody structures. At the bottom, it allows for filtering by properties such as species or sequence. Here we find over 8,000 structures, all aligned, superposed, and annotated consistently. It happens to be the same antibody database that we used earlier in the Antibody Modeler to identify the framework and the CDR templates when we generated our mouse wild type. Beyond searching for CDRs, VL, VH, or antigen sequences, we can also search for 3D motifs, characteristics, and other properties using the selection language. It also contains a browse panel, so if I just click browse, that allows us to browse through the structures within this database which have been superposed relevant to my model. If I wish to keep one of these structures, I can click keep, which would send that directly to the Mo system, which I find to be a useful feature. But today I'm going to just close that, and actually what I want to do is analyze the statistics across the antibody family to find a suitable mutant for our serine. So for that, I'll click the stats button, which opens up the statistics panel of the antibody database. So this currently is showing me the distribution of the species found across the entire antibody database. In this case, though, we know that we're going to want to search for humans. So I'll just go ahead and refine our search for human, click search, and we see now that our statistics panel has been updated to reflect the statistics across only the human antibodies. Beyond searching for statistics of the species, there are different parameters or classifiers that we can search, and I'm going to be using UID, which provides residue distributions for each position, this time using only data for the human antibodies, which reflects our filtered search list. By default, this panel opens up showing us the distribution shown in position 1 of the light chain. To look at the position of interest, I can either type it into this box or I can use the arrows to click through the different residue positions. However, because I've got the serine selected in the main Mo window, I can go ahead and click this target button, which retrieves the residue distributions for that selected residue. So here I see that for position 115 in the sequence, proline is the most prevalent residue at that position. That tells me that proline could therefore be used to eliminate the glycosylation site and gives us a good chance at maintaining stability and preventing immunogenicity, since it's common at this position in human antibodies. Now that I've identified the residue that I want to mutate to, I'm just going to close our statistics panel. I'm going to close the project search panel. And to make this single point mutation, typically I'd have just two options. The first would be to use the builder, which is well suited for making a single point mutation such as this. An alternative approach would be to use protein design using residue scan, which is well suited for when I want to make multiple mutations, or perhaps analysing the impact of a mutation upon affinity or stability. In our case, though, I'm going to be exploring a third option, and that's to look at the 14 candidates that we just modelled, and we'll see if any of these happen to exhibit a lower hydrophobic patch and or feature that glycosylation site that we just identified. So I'm just going to go ahead and close the database viewer that contains our wild type, and I'll actually close our system now, and we'll go ahead and have a look at the database viewer which contains our 14 candidates. So it's the same format as we found for our mouse wild type. You'll notice that the wild type has been included as part of this modelling process. We identified that that had relatively poor developability. The variants that have been tested here tended to behave better with respect to chromatography. Having the wild type is important because it establishes a baseline to which we can reference the other structures. Within the database viewer, we find other fields such as the templates that have been used to create these models. However, to help analyse the 14 candidates with respect to good developability, we're going to calculate the protein properties so that we can rank order our antibody series. So I'll just go ahead and open the protein properties panel, which comprises two main tabs. We've got the property calculation tab, which contains a list of just over 100 possible descriptors. The 28 most commonly used have been selected by default, which include the calculation of hydrophobic patch areas, charge descriptors, and PI predictions. I'm just going to go ahead and start that calculation. The second tab is the property viewer tab. That will become, it's grayed out currently, but it will become visible once the calculation is complete, and it will be populated with the resulting values and a set of per residue properties such as surface areas and residue pKa's. Today we're calculating the properties using a single static model. However, there is a sample option. So if I toggle on sample, that would have allowed exploration of conformational and protonation samples for each model, and provides versions of these properties that have been averaged on the conformational ensembles. This tends to give better correlation to experiments, but it is a longer calculation. And for that reason, we might want to consider using a cluster or the cloud. So for that, we can use this batch button, which takes the input parameters that have been specified here. And instead of clicking calculate to run them locally, we click batch to send that run file to the cloud or a cluster. I see the results are ready, but I just want to go back to the property calculation tab just to point out a few of these default descriptors that we're calculating. So one of these would be the ensemble net charge. It's an important parameter when it comes to analysing viscosity. Another important descriptor would be the area of the hydrophobic patches near the CDRs. We've already spoken about this being an important parameter, which often correlates with experimental aggregation and solubility data, whereby the lower the value, the better the developability. So the results are ready, so let's just click across to the Property Viewer tab, and here we find those results in the viewer mode. You'll notice that some of these descriptors have a plus next to them. So for example, this one, residue ASA, which stands for accessible surface area. So if I click on the plus, it then gives me a readout for each of the accessible surface areas of each of the residues within our model. Often though, I prefer, if I'm looking at residue information, to toggle on this residue table, which gives me a single view with a complete overview of all of the residue properties that have been calculated. However, these results have also been written to the database viewer. So what I'm going to do actually is just close protein properties, and if I scroll along, I see that those properties have been written here as well. So you'll recall in the database viewer that the first line was our wild type, which had relatively poor developability. The variants that were tested generally had better chromatographic behavior, and we can see here that the hydrophobic patches near the CDRs tends to match these experiments, where the mutants that had lower HIC retention times also have lower hydrophobic patch areas. So what we're going to do next is load in the wild type and one of these candidates that has the lowest hydrophobic patch area, and we'll analyze these hydrophobic patches in 3D to see if we can explain their differences. So what I'm going to do is sort according to one of these fields within the database viewer, so we can sort by any of these. So I'll just go ahead and sort descending. It'll ask, are you sure you want to do that? Yes, I am. So now we see that the hydrophobic patch area now decreases. So what I'll do is go ahead and select the wild type. I'll select the one with the smallest hydrophobic patch, and what I'll do is send those selected models to Mo. We see that they appear in the main Mo window. It looks as one, but there's actually two. It's just that they've been superposed and annotated consistently with the Mo project antibody database. We can analyze these in 3D, but we can also analyze them in terms of their sequence. So in the sequence editor, we find the wild type, we find the mutant. So to help me analyze those, I'm just going to go ahead and select the mutant, and what we can do is color those residues by similarity, whereby blue represents the residues that are most similar, red represents those that are most distant. So I see here that we've got a red residue or a pink residue, but this isn't in a CDR. Perhaps of more relevance is this mutation here that sits directly in a CDR, and this mutation could well be responsible for the decrease in the calculated hydrophobic patch descriptor. We can also analyze this in 3D, so if I go ahead and calculate protein patches as we did, this time for the two models. So just to recap, that the protein patches are showing us the regions with excess charge or hydrophobicity. Now these are superposed, so we can use the system manager to control these. The first line here would represent our wild type, the second line for our mutant. So if I just toggle on and off the wild type, you'll see that that mutant doesn't have the or at least it's significantly broken around that region there, disrupted by the residue, that one residue that we find mutated. So that brings us to the end of today's session. Today we've been modelling and analysing an antibody to find liability motifs and the risk of aggregation using our selector. We then compared the wild type to other antibodies using the project search. Upon finding that our wild type had poor developability, we then modelled 14 candidates. We analysed their properties, we rank ordered them, and we selected the most promising candidate with a lower hydrophobic patch area, which we hope would have improved developability experimentally. I hope that you've learned a lot today. If you have any questions, please do reach out to me at support@chemcomp.com.